Good morning all. Today we are going to study about the topic DNA as genetic material. Before going to the details about the topic, it is necessary to know about what is a genetic material. A genetic material is a substance that carries the encoded biological information about the structural, functional, developmental and behavioral properties of organism. It is also an agent that transmits coded biological information from parent to progeny. All of you know that in most organisms DNA is the genetic material, but in some viruses RNA serves as the genetic material. Then coming to the properties of the genetic material. First one, it has the ability to store and transmit biological information in a stable and coded form. Second one, it has the ability to replicate with high fidelity to produce identical functional copies. Third one, it has the ability to distribute its copies equally from parent cells to daughter cells with extreme accuracy and minimal error. The DNA is highly stable, that is, it has high physical and chemical stability. It helps to ensure genetic constancy in organisms. Last one is that it has the potentiality to genetic variations. These genetic variations leads to genetic diversity among organisms. And coming to the topic, DNA as genetic material. DNA was first identified by Frederick Nisha in 1869. He isolated DNA from the blood cells and termed it as nuclein on the ground that it is a constituent of nucleus. Later, Richard Altman designated it as nucleic acid since it was acidic in nature. Then Nisha strongly believed that biological information might be passed from parent cells to progeny cells in the form of a chemical code. Then Oscar Hedwig in 1884 was the first to suggest that nuclein is the substance that is responsible for the transmission of hereditary characters. And on the basis of several experiments conducted by different scientists, later it was proved that DNA is the genetic material. Then coming to the evidences which prove DNA as genetic material. The first evidence came from the classic work of Frederick Griffith on bacterial transformation in 1928. After Griffith's experiment, Oswald Avery, Maclean McCarthy and Colin MacLeod experiments on transformation proved that DNA is the genetic material. And the final experiment is Hershey and Chase experiment on T2 bacteriophages. And coming to the evidences from the study of bacterial transformation, or it is otherwise called the Griffith's experiment. The first clue about the fact that DNA is a genetic material came with the demonstration that DNA was responsible for a phenomenon called the transformation. The phenomenon was first observed in 1928 by Frederick Griffith. Then Griffith had succeeded in isolating different strains of Streptococcus pneumoniae. There are different types of Streptococcus pneumoniae, type 1, type 2 and type 3. And there are mainly two different forms of this strain. They are virulent forms and non-virulent forms. In the case of virulent forms, each bacterium is surrounded by a polysaccharide coat, which makes the bacterial colony appear smooth when grown on agar plate. These forms are referred to as smooth, and this can be represented by the letter S. Then Griffith found that some of the virulent forms occasionally mutated to non-virulent forms, which lacks a polysaccharide coat and produce a rough appearing colony. So they are called rough colonies, and they are represented by the letter R. Then coming to the experiments conducted by Griffith, in the first set of experiments, he injected living 3S bacteria. 3S means type 3 and S means most strains, they are virulent forms. So he injected the living 3S bacteria into mice. Then the mice developed pneumonia and die. He found large amount of type 3S bacteria in the blood of the mice. In the second set of experiment, Griffith injected type 2R bacteria. Type 2R means they are non-virulent bacteria. He injected this type 2R bacteria into mice. The mice lived and no bacteria were recovered from the blood. When he injected large amount of heat-killed type 3S, you know that type 3S bacteria are virulent forms, but here he used heat-killed type 3S bacteria into mice. Then the mice lived and no type 3S bacteria were recovered from their blood. In the last set of experiments, he injected small amount of living type 2R bacteria along with a large amount of heat-killed type 3S bacteria into mice. Then the mice became infected with pneumonia and died. Remember that? Here he used heat-killed type 3S bacteria and living 2R bacteria. But the mice get infected with pneumonia and died. Griffith examined blood from the hearts of these mice. He observed live type 3S bacteria. What may be the reason for that? So there are certain interpretations about Griffith's experiments. The first interpretation is that it could have been the case that he had not sufficiently sterilized the type 3S bacteria and thus a few live bacteria remained in the culture. But Griffith knew that there is no possibility for that because he had used only heat-killed type 3S bacteria in the control experiment and they never produced pneumonia in mice in the control experiment. The second interpretation is that the live type 2R bacteria had mutated to the virulent S form, but there is no chance for such type of mutation. Normally the type 2R bacteria are mutated to type 2S bacteria, but here he observed a live 3S bacteria in the blood. So there is no chance for such mutation. This is the diagram showing Griffith's experiment. Here, this is the first set of experiment. Here he used rough strains and he injected the rough strains into mice. Then the mice lived. In the second set of experiment, he used the smooth virulent forms. Here the mouse dies. In the third set of experiment, he used the heat-killed smooth virulent forms. Here the mouse lived. And the third set of experiment, he used both the rough non-virulent forms and heat-killed smooth virulent forms. Then the mouse dies. From these experiments, Griffiths came to the conclusion that the type 2R bacteria had somehow been transformed, acquiring the genetic virulence of the dead 3S bacteria. This transformation had produced a permanent genetic change in the bacteria. But Griffith didn't understand the nature of transformation or how this transformation takes place. He concluded that some substance in the polysaccharide coat of the bacteria might be responsible. He called this substance as transforming principle. These are the conclusions of Griffith's experiment. Then coming to the identification of the transforming principle. What is that transforming substance or what is that transforming principle? After Griffith's experiment, Avery, Colin MacLeod and Maclean McCarthy succeeded in isolating and purifying the transforming substance. They showed that these transforming substances have the chemical composition closely matching that of the DNA and quite different from that of protein. They used different sets of experiment they use the enzymes which can digest proteins, and also the enzymes which can digest RNA, and also the enzymes which can use DNA. When he used the enzymes such as trypsin and chymotrypsin, which can break the protein, had no effect on the transforming substance. But when they used the ribonuclease, that is the enzyme that destroys RNA, it had no effect. But when they used the enzymes capable of destroying DNA, they can eliminate the biological activity of the transforming substance. This is the diagram showing the experiments conducted by Avery, McCarthy and MacLeod. Here you can see that the type 3S bacterial filtrate was there. They treated this type 3S bacterial filtrate with the RNAs, protease and DNAs. Then these enzyme treated filtrates were added to the different cultures of type 2R bacteria. And here we can see that in the culture treated with the protease and RNAs, type 3S and type 2R bacteria are there. But in cultures treated with the DNAs, only the type 2R bacteria are there. So from these experiments, it is clear that DNA is the genetic material. Then coming to the Hershey and Chase experiment, it is another evidence implicating DNA as a genetic material resulted from the study of the T2 virus conducted by Alfred Hershey and Martha Chase. T2 viruses are bacteriophages that infects the bacterium Escherichia coli or E. coli. In their experiment, they designed a series of experiments to determine whether the phage protein or the phage DNA is transmitted in phage reproduction. To follow the path of protein and DNA, they used radioactive forms or isotopes of phosphorus and sulfur. But what is a radioactive isotope, or why these radioactive isotopes are used? The radioactive isotopes can trace the path of a particular molecule. A radioactive isotope can be used as a tracer to identify the location of a specific molecule, because any molecule containing the isotope will be radioactive and therefore easily detected. Here you know that DNA contains phosphorus but not sulfur, but protein contains sulfur but not phosphorus. So Hershey and Chase used P32 to follow the phage DNA during reproduction, and they used S35 to follow the protein. Next is the steps involved in the experiment. Hershey and Chase grew one batch of E. coli in a medium containing P32 and infected the bacteria with the T2 phage, so that all the new phages emerged from the bacterial cell have DNA labeled with P32 because the medium containing P32. In the second set of experiment, they grew a second batch of E. coli in a medium containing S35 and infected this bacteria with the T2 phage. So all these new phages emerged from the bacterial cell have protein labeled with the S35 because they are cultured in medium containing S35. Then in the third set of experiment, Hershey and Chase then infected separate batches of unlabeled E. coli with the S35 and P32 labeled phages. Then after allowing time for phages to infect the cells, they placed the E. coli cells in a blender and sheared off the empty protein coats. These protein coats are called protein ghost from the cell walls. They separated out the protein coats and cultured the infected bacterial cells again. When phages labeled with 35S infected the bacteria, most of the radioactivity was detected in the protein ghost and little was detected in the cells. When new phages emerged from the cell, they contained almost no S35. These results indicated that the protein component of a phage is necessary for infection. It does not enter the cell and is not transmitted to the progeny phages. Then in contrast to this, when Hershey and Chase infected bacteria with the P32 labeled phages and removed the protein ghost, the bacteria were still radioactive. After the cell lysed and new progeny phages emerged, many of these phages emitted radioactivity from P32. It proves that DNA from the infecting phages had been passed on to the progeny. These results confirmed that DNA is a genetic material, not the protein. This is a slide showing the experiments conducted by Hershey and Chase. Here you can see that the bacterial cells cultured in medium containing S35, and here the bacterial cells cultured in medium containing P32. Here the unlabeled bacteria are infected with the S35 labeled phages, and here it was infected by using P32 labeled phages. After allowing time for infection, these bacterial cultures were placed in a blender, and after that they are centrifuged, and you can see that here the protein ghost or the protein codes are S35 labeled and the bacterial cells have no radioactivity. But here the protein codes have no radioactivity, but the bacterial cells have radioactivity. Here the new progeny phages emerged from the bacterial cell by the lysis of the bacterial cell wall. They are not radioactive, but here the new phages emerged have a radioactivity. This experiment also proved that DNA is the genetic material. Then coming to the summary of the topic DNA as genetic material. In this class you have studied about what is the genetic material and what are the properties of a genetic material, how DNA is called genetic material, and what are the experiments proved that DNA is the genetic material. So once again, genetic material is a substance that carries the encoded biological information about the structural, functional, developmental and behavioral properties of organism. It is the agent that transmits coded biological information from parents to progeny. The classic work of Frederick Griffith on bacterial transformation, Oswald Avery, McCarthy and MacLeod's experiment on transformation, and Hershey and Chase experiment on T2 bacteriophages have proved that DNA is the genetic material. It is all about the topic DNA as genetic material. Thank you.